Thanks for joining the presentation. As Ant mentioned, my name's Tamsin. I'm Market Development Manager in Illumina, and my role is to really enable the use of our technology in new and novel ways. And it's quite a cool role to have. But we've been working a lot in COVID in the last couple of months, and I just thought I would share some of the work that's been happening in Australia and across the globe in this space, with a slight touch on Illumina tech as well. So one obvious example of how NGS technology can be applied in new and novel ways is in the field of virology. And this technology is being used in a myriad of ways, including the discovery and characterisation of novel viruses. And in other ways as well, we are working with public health labs to use NGS for the surveillance of pathogens, including viruses, but also bacteria and other pathogens as well, to understand how they evolve, how they behave, and how they move between different locations across the globe. We've also seen NGS used in the development of molecular diagnostic tests. And as has been quite topical in the last couple of days, NGS has actually played a really pivotal role in providing the foundation of a lot of sequence context for the development of vaccines. Illumina has worked closely with Moderna in the development of their vaccine, providing sequence information there, and other therapeutics to guide antiviral treatments. Talking about COVID, so we'll take a brief step through the COVID timeline and walk through the COVID timeline of the pandemic. So on December 8th, there was a first documented case of a patient developing respiratory illness symptoms, if I could say that correctly, respiratory illness symptoms in Wuhan, China. And over the Christmas-New Year period, the China authorities alerted the WHO about these respiratory illness cases. But at the same time, Illumina, with the China CDC and the Shanghai CDC, began to work to use some molecular techniques to actually try to unveil the sequencing of this novel virus that was not responsive to existing therapies. And this was published on the 7th of January, where a new novel coronavirus causing COVID-19 was actually noted in the public literature. The virus spread very rapidly, and the first case was recorded outside China, actually in Thailand on January 13th. And a lot of, obviously, movement orders were placed in Wuhan to restrict movement of people across the country and across the world. Moving forward, on March 11, the WHO actually declared the SARS-CoV outbreak to be a pandemic. And just stemming through, we've all lived through the various restrictions, movement restrictions, lockdown, changes in social practicing. But just to put in context, today we're at over 50 million cases of the disease, with sadly more than 1.25 million deaths. This has actually provided a really unique advantage into fighting COVID-19. I hope to show you how we use Illumina technology with our Chinese researchers to identify the virus that was causing these respiratory illnesses, but how the use of sequencing extends beyond just that discovery work to how we can understand transmission routes of the virus between countries, between cities, even within hospital settings. How we can actually use the sequence information of the virus to look at how its sequence is adapting as it spreads, perhaps making the virus more virulent and with larger transmission routes. We can use we would briefly discuss on the role that sequencing can play in identifying targets for therapies or diagnostics, but today I'm actually going to talk very, very briefly in the presentation about how sequencing can also make sure that diagnostics, in particular molecular diagnostics, are indeed performing as they should be. And finally, we'll just very briefly touch upon how surveillance is really required to understand the role of co-infection in COVID diseases as well. I am a Illumina employee, so I need to talk about how we're going to sequence the SARS-CoV-2 genome. And there's three main techniques that we could use here. The first is using a brute force technique, where we just literally take all total nucleic acid substrates within a particular sample, could be a sample in the case of COVID-19, and sequence everything, and then use bioinformatic approaches to actually characterize the nucleic acid species that are present in that sample. Second, we can use a slightly more selective approach and enrich the viruses. Most common ways to use a biotinulated probe to pull out just sequences that we're interested in. And we have a range of panels that can pull out sequences for a whole suite of pathogens, both bacterial and viral, to really understand the true nature of a nucleic acid subset in a sample. Or we could be quite cost-sensitive, time-sensitive, and just amplify just the virus itself. So just amplify the SARS-CoV-2 virus, ignore everything else, and get this sequence information directly from that, and use that sequence information in our study. And the amplification method to date has really been the most popular method that's been applied in the community as such. At Illumina, we quickly formed a task force team and really tried to position some workflow solutions to enable their customers to perform this sequencing really quickly. So we do have a workflow for each of the three sequencing protocols I mentioned earlier: a metagenomics workflow, which can use our NextSeq system, an enrichment workflow, which utilises a new kit called the respiratory virus oligo panel, which I'll touch on later, and also a anticon-based workflow, which is a COVID-seq test. Now, this test has EUA approval from the US FDA, but in Australia, we actually use this kit in research use only mode. And also this kit can also be run on the NextSeq. We were really fortunate to actually get the support of our bioinformatics teams when the COVID outbreak really started to gain momentum, and get their buy-in to develop some workflows and some pipeline workflows really quickly to enable customers, to analyse the data in a quick, really accurate manner, and then more importantly, or equally as importantly, share that data with the broader scientific community. So we developed three pipelines to assist with data analysis, be the Dragon enrichment pipeline, the Dragon metagenomics pipeline, or the Dragon RNA pathogen detection pipeline. And we also the team also developed a couple of pipelines to enable customers to automatically upload data to GISAID, which is a global repository of sequence information that is publicly available, and also directly upload data to the short read archives as well, just removing some bottlenecks for our customers. So where we've identified some ways in which we can help the scientific community, we've really tried our best to do things to remove barriers to enable the science to proceed as quickly as possible. You'll be relieved to know I'm not going to be covering technical details about each of these products today. If anybody would like information, Answer Guru, or please feel free to contact myself or your local Illumina representative. We'd be more than happy to discuss any of these workflows in more detail with you. But now we'll actually move on to how the NGS is actually being used in the community to actually guide and fight against COVID-19. So over to China, again, as I mentioned, we were engaged with the Shanghai and China CDCs to actually help identify a novel virus that was causing respiratory illness. Here, a 41-year-old male working in an indoor seafood market in Wuhan, China, presented with clinical symptoms. Several tests were performed, but these were all negative. At this point, we were called in and with China CDC team putting a lot of effort into this work, actually started to see and use NGS to identify this novel virus that was causing the disease. So we extracted total RNA from a bowel sample and actually performed 2 mini-seq and then some confirmatory NextSeq runs to actually analyse the data, sequence the data. The largest contigs assembled from this data were then blasted against databases, which came back with a close match to a bat SARS-like coronavirus that was also previously identified in China with a nucleotide of identity or similarity of 89.1%. This sequence was submitted to GenBank and designated as WH1 human coronavirus 1. Oh, it's already moved on. I've got some photos from our Illumina family photo album, just showing Illumina personnel and China CDC personnel actually performing the work of preparing the libraries to run these samples on the NextSeq system. And you can see the NextSeq system in the lower left-hand corner showing the 2019 NCOV run actually performing the sequencing of that metagenomic sample on the sequencer as it was taking place. We always need to make sure our troops are kept happy and not hungry. So lunch runs always are an important issue, tasks that Illumina people are more than happy to deal with as well. So moving on from that, we have the foundational genomic sequence of the virus, or of the potential virus that is causing this illness. And one of the first questions that always comes out is well, where did this virus come from? And I know that you guys have heard a lot of this information before, but I think it's really important again to note just the power of NGS enabling us to compare the genome sequence of this novel virus, which is designated SARS-CoV-2 here, to other viruses to identify similarities and use that as a stepping stone for guiding future public response and treatments and therapeutic developments. So the SARS-CoV-2 is actually the 7th identified coronavirus in human. There are 4 which are known to cause less severe symptoms consistent with common colds. But there are 2 other ones, the SARS-CoV and MERS-CoV, which are actually cause more severe illnesses. So as you remember, the SARS-CoV-2 was from an outbreak in 2002, severe acute respiratory syndrome in Asia, and the MERS is Middle East respiratory syndrome, which was identified in 2012. Both MERS-CoV and SARS-CoV are beta coronaviruses, and they have their origin in bats, but different intermediary hosts before transmission to human. Recent emergence of the SARS-CoV-2 virus is likely to be derived from an animal reservoir as well, although we still haven't got down to the nitty-gritties of which actual animal that was in the first instance. And I'm not sure if any of you have been following news, but they've also been recently recognised in Europe some transmission of the virus to and from minks. The SARS virus is being transmitted from human to minks and minks back to humans. So it's obviously a very highly zoonotic virus that can be transferred easily between animal and human. And I know that you've all seen multiple pictures of the SARS-CoV-2 virus, the virus that causes COVID, so I'm not going to go into a lot of detail here. Just to say that of viruses, it has a very large genome, approximately 29 kb in length. This genome is encapsulated by a glycoprotein with spikes on the outside to help aid binding to host cells. So we know the sequence, we know where it comes from, but how can we actually accelerate efforts to really generate SARS-CoV genome sequences to understand how the virus is behaving, how it's evolving, how it's transmitted. And a lot of efforts went on really quickly in the pandemic to really define methodologies that could be used to accurately and quickly sequence the coronavirus, to use this information to guide and help inform public response. So in the UK, the COG UK consortium, COVID-19 Genomics UK consortium, was set up with the aim of sequencing approximately 180,000 SARS-CoV-2 genomes. There are a multiple number of members of this consortium, including Illumina and Arctic. So the Arctic network is the network that primarily developed a really nice amplicon-based assay for sequencing the SARS-CoV-2 genome, which is comprised of 98 overlapping amplicons to span the entire genome. Illumina, as a part of this network, then worked with the Sanger Institute to really refine down this Arctic protocol, make it high throughput, make it robust and consistent on Illumina tech, so that it could be used around the world by scientists to accurately and quickly sequence the SARS-CoV-2 genome. So we developed the COVID-seq assay with the first aim for surveillance. However, upon understanding its surveillance, we also realised that this assay has the ability to test for the presence or absence of the coronavirus in patient samples. And over in the UK, we took that one step further and obtained EUA approval from the FDA for the COVID-seq assay for detection of the SARS-CoV-2 virus. That approval was obtained in approximately August this year. I promise no text guides, but I promise I'll be quick on this just to walk through a brief walkthrough of the COVID-seq workflow. It is an amplicon-based workflow, so very fast, very simple. We start with some NPOP swabs, extract the RNA from those swabs using extraction material from our partner Zymo, and this is an automatable process. Then go through cDNA conversion, moving on to PCR, which is that Arctic-based protocol as we've mentioned previously. So there's 98 amplicons tiled across the genome. We also have 11 additional internal controls, which are human amplicons. We move into library prep and pull our samples, and then we can run them on either a NovaSeq or a NextSeq system. NextSeq system can take 384 samples per run. A NovaSeq can take 3,072 samples per run. And the overall turnaround time is about 24 hours from sample to solution. Whoops, sorry, I went the wrong way, sorry. So as you may have seen in the previous slide, or a couple of slides ago, COVID-Seq has been developed with both testing and surveillance in mind. So I just wanted to make sure I was clear in how I was using these words in context with the SARS-CoV-2 and the COVID-Seq assays. So diagnostic testing is aimed at an individual. So aimed to provide a yes/no answer, is the virus present or absent, so that appropriate treatment can be provided. Surveillance, on the other hand, is looking at a more population-based analysis to really track the path of endemics, to understand transmission and the rate of viral evolution. So we'll be focusing mostly on surveillance from now on, not diagnostic testing, as that is really exclusively available in the US. So surveillance is comparing the genome sequences of multiple SARS-CoV sequences from across the globe to really understand the behaviour and evolution of this virus over time. And as I touched upon just before, GISAID is one of the central repositories for SARS-VOT sequences, and it's got sequences deposited from all over the world. And recent stats, it had approximately 160,000 SARS-CoV-2 genome sequences. Australia has definitely pulled its weight, and kudos to the Australian scientists for really doing a great job in sequencing quite comprehensively patient samples of COVID-19 patient samples, approximately 14,000 of the SARS-CoV-2 genome sequences in the GISAID database are from Australia. And this heat map of the globe, which shows the number of sequences submitted to GISAID per 1,000 cases, really sort of demonstrates how Australia has proportionally made a really strong contribution to the GISAID database. So we're in good partnership with New Zealand, with Norway, with the UK, Vietnam, and even the Democratic Republic of Congo. And just diving in a bit more to some of the work that we've been doing in Australia, there's some really beautiful publications that have been coming out from Australia, multiple publications on COVID-19. But these two publications shown here have really shown nicely how surveillance can be used to understand the viral behaviour within a community population. So to really understand transmission routes and to combine genomic information with epidemiological information to really help form and guide public health response to this outbreak. So these are two beautiful publications from Rebecca Rocket at Westmead Institute and Torsten Seaman at MDU in Melbourne that I would strongly encourage you all to read, to really understand the breadth and just the depth of work that's been undertaken in Australia with performing genomic surveillance to really understand and help guide our strategies moving forward. A couple of points that they have noted is that there are multiple importations of the SARS-CoV-2 virus into Australia, which we pretty much knew, but that's great to have it confirmed in a proper scientific manner, a rigorous scientific manner. They have also identified some viral transmission events within a healthcare setting and demonstrated the utility of genomics in infection control. And there was such promising work coming out from these sites and sites all around Australia, not just to isolate these sites, but there are multiple sites around Australia doing this great work, that the government decided to release some funding after much lobbying really from the Communicable Disease Genomic Network and Illumina as well, to enable the detailed tracking of COVID-19. So around June time, the Australian government announced funding for a grant specifically focused on the need of pathogen genomics and SARS-CoV pathogen genomics in Australia. We at Illumina are very privileged to be involved in this grant, and we've enabled this with some sequencing reagents and consumables with the aim of really contributing towards our understanding of the virus. These labs across Australia are performing best practices, but to get them standardised around the country is really another goal of this project, and also to use these standardised methods to understand the phylogenetics of the virus and understand its reproductive rate, And ultimately, we hope a lot of the work that's undertaken in this project will really help guide future pathogen genomic frameworks and help guide how Australia can actually have a strategy in place that should outbreaks occur again in future, that we're actually appropriately positioned and adequately funded and ready to go at the get-go. So as some more pictures from the Illumina family photo album. On the upper left-hand side, we have some pictures of displaying a family of Illumina systems getting ready to sequence some COVID samples in the lab. We also have Gavin, one of our awesome application scientists, installing a NextSeq 2000 system down in Melbourne. And the bottom system is maybe Rebecca Rocket's finger or maybe someone else's finger. This is actually taken from some great footage that was covered by 60 Minutes on Channel 9, just to discuss how genomics is actually being used. So we're really starting to get some airtime and starting to get more visibility, because it actually is really playing a very crucial role in this COVID-19 outbreak. Moving on, just over the next few slides, last few slides, I'd just like to discuss how surveillance can actually help inform response and guide public health response to outbreak, and how we can guide our recommendations to the general broader community. So these are a couple of examples again from the COVID-19 consortium in the UK. The first one is looking at, if you remember, you may remember back, they committed to sequencing 180,000 virus isolates. But we have a first analysis of 20,000 viruses, which was sequenced up to May this year, with the aim of identifying patient zero. Where did the virus come from? And the study actually found that there was no patient zero. In fact, there were 1,356 separate routes of entry of the SARS-CoV-2 virus, more than 10 from more than 10 countries. From this, the UK government actually, quite belatedly, but did implement some responses to this and counselled international travel and began to impose traveller quarantine measures to restrict the transmission of the virus further. So there's one example of how surveillance can really help guide public health response. The second example is more of a hospital-based sample, where there were 6 patients who spent time in a non-infectious disease ward, which we called Ward A, who all contracted COVID-19. Here, genomic information combined with epidemiological information was able to enable scientists to determine that the virus was actually transferred within the ward, enabling, again, a response to be immediately actioned. And in this case, the ward was closed, deep sequencing was performed, the patients were monitored, and additional safety precautions were put in place, which is a really great measure. Another example of how global surveillance can really help alert this is by monitoring the sequence of the virus and identifying new mutations in the virus which could potentially lead to changing function. The first really characterised example of this was the A to G mutation, the RNA sequence of the genome, which causes an aspartate to glycine substitution in the spike protein. This is translated to a protein sequence which describes the function of the virus. This is an example of an isolate in the SARS-CoV-2 where a single change in the genome can immediately reveal the fact that the spike glycoprotein 2 has changed. And there's been lots of work into the role or the change in function of the spike protein upon acquisition of this mutation. There are multiple hypotheses that it does change the virulence, that it doesn't change the virulence, but there is definitely the potential to have a change in function of the spike protein upon this mutation being acquired. And in fact, this mutation has become more dominant over time. If you have a look on the right-hand side of this slide, you can see that around February time, on the left-hand side of each of the 6 locations we're looking at, the D614 variant was more common. And moving over time, we see an increase in the prevalence of the G614 variant of the virus, particularly in places like South Korea and Australia. And one finally, sorry, I've got 2, not 1. second last, penultimate example of how genomic sequence and surveillance sequence can actually help guide our understanding is through the generation of multiple sequences, we can help understand if it will have an impact on the efficiency and sensitivity of molecular diagnostics. This is a image I grabbed from a recent preprint from bioRxiv, but it really demonstrates the point quite nicely in the sense that analysis of the SARS genome may identify new mutations that lie in the same location as primer binding sites for existing qPCR molecular diagnostic assays. And there is a potential then for this to affect the performance of the assay. There have been multiple reports of different assays being affected in sites, but this particular publication or preprint actually identified 10 potential assays that could be affected by the presence of mutations underneath their primer binding sites. So it's really important to keep genomic surveillance, have ongoing genomic surveillance, to keep aware of the strains that are in circulation within particular communities at time, to make sure that molecular diagnostic assays are indeed relevant and are indeed accurate and are indeed giving accurate diagnoses of patient samples. So just a nice example of why it's really important to keep genomic surveillance going as this pandemic continues to evolve. And one last thing, just one last sort of take home to think about is that in a certain proportion of cases, we see that COVID-19 is not the only respiratory pathogen that has infected a patient sample. And there's a really nice study that was done at Stanford Health Centre in about May this year, where they analysed 1,200 different isolates from a Northern California community to look at the presence of other pathogens, in particularly viral pathogens, so other viruses, in conjunction with the SARS-CoV-2 virus, so trying to identify co-infections. And in 22% of cases, the SARS-CoV-2 virus was actually co-infected with other viruses such as influenza and rhinovirus. So the role and the impact of these co-infections, or what it has in patient, sorry, we're going too far ahead, what it has in patient response remains to be determined. But, you know, we need to understand how co-infections actually affect the patient's ability to respond to recover from the SARS-CoV-2 infection. And this study actually used our respiratory virus oligo panel, whose contents are shown on the left-hand side, which include coronavirus and the SARS-CoV-2 virus. And one of the reasons why there's a couple of reasons why we're particularly interested in this is, first of all, it's going into winter time. We've all seen a spike in cases across Europe and the US, and we think this could really help give us further resolution to the infection rates of COVID and the transmission of the SARS-CoV-2 virus across these areas, and help understand why perhaps host responses varies between individuals, in addition to host genetic factors as well. From an Illumina standpoint, you know, looking at multiple co-infections really aligns nicely with NGS technology, as it enables us to get really detailed, accurate resolution across a broad, comprehensive panels in a single assay. So we'll be investing more time and more effort into developing panels, enrichment panels, to assess the presence of multiple pathogens, in particular viruses, in a number of disease states moving forward from here. And just to wrap up, I think one of the main sort of take-home messages from me, having worked quite extensively in COVID-19 and the COVID-19 outbreak since January, is really that we need to have a global genomics platform for pathogenomics. We need to have greater alignment. We need to have frameworks in place so that all countries are prepared and when an outbreak will occur, because another outbreak will occur. And as you can see from this slide, you know, there's been multiple outbreaks that have occurred since 1980, and these can have a devastating or a significant impact on economies and on societies as a whole. And to know that, you know, the estimated impact of COVID-19 is $18 to $15 trillion in the US is a really significant point. We haven't even touched upon antimicrobial resistant infections as well, and the role that they can have. But to take it all to that holistic level, and that we need to get this global alignment, and initiatives are beginning to start to take place towards this end about how we can work together to really enable people to share data, to get frameworks in place, to learn from countries like Taiwan, Hong Kong, Singapore, who had frameworks in place before COVID-19, and we're really positioned to act swiftly and act in a really considered, intelligent way to the outbreak, and the impact that has on their communities has been really, really positive. So we hope that in the future, the world will start to, we have a utopian view that greater alignment and greater collaboration across the globe in pathogen genomics will evolve and will enable us to be better prepared when future outbreaks occur. And finally, Illumina is committed to enabling and doing all we can in the fight against COVID. It's been great to see our technology being used across the country, and we're continuing still to this day to develop products to provide support and training to our internal teams and to our customers to enable them to perform this really important work, and to, where possible, remove all barriers to enable you to perform this important work. And we are in it for the long haul. So with that, I'll turn back to you, Ant, and thank you so much for your time.